is the vaccine, and what is WHO's comment on that?
Do you want me to take that?
Dr. Simao?
Margaret?
Yes, please go ahead.
Yes, thank you very much for that question. The first thing is that WHO, of course, welcomes all vaccine development programs around the world. And we're very encouraged by the fact that there are so many vaccine development programs and over 30 candidates now in various stages of clinical development in phase 1, 2, or 3 clinical trials. We have also put out quite early on in May this year the criteria for what would be considered a safe and effective vaccine. It's called a target product profile, and it describes the kind of sort of a benchmarks that we would like a vaccine to meet. So a minimum of 50% efficacy in preventing infection with a lower bound of at least 30%. So a vaccine that offers at least 30% protection at the population level, and of course is safe. Now, safety, again, is assessed short-term, but also needs to be assessed longer-term because there are some side effects which you only pick up later on. So that is why it's so important to have these clinical trials conducted according to the standards and norms so that the data can then be examined by the experts before a decision is made on whether or not this vaccine should be licensed. So we have started discussions now with the authorities in Russia to learn more about the vaccine candidate, and we've requested for the data on efficacy and safety. We understand that it's gone through some preliminary human studies and that it is about to get into a phase 3 clinical trial, which will really be the test of efficacy. So we look forward to discussing with the Russian authorities as well as seeing the data that is available so far and then having a dialogue on what the further needs should be and how further studies would need to be done. And this is why we're also promoting the idea of a solidarity vaccine trial, where many different manufacturers, developers, countries can participate, both as trial sites to test different vaccine candidates, but also to provide the vaccine candidates into this large global multi-arm adaptive clinical trial that we think will be both a cost-effective and an efficient way of testing as many candidates as possible in the shortest period of time. Thanks.
Thank you very much, Dr. Swaminathan. So we have no more comments in the room. I will now move to Christine from ABC America. Christine, please unmute yourself and go ahead and ask your question.
Thank you. I wanted to ask about monoclonal antibodies. Could you speak to what they are, how they can be used, and limitations to getting the drug to the population at large? Because it sounds like a groundbreaking candidate therapeutic that may play an important role in helping curb the pandemic. Thank you in advance.
Thank you, Christine.
I could start, Margaret.
Yes, good. I was going to say it's for you.
Might want to add. So monoclonal antibodies are basically highly specific antibodies which act by preventing the virus from binding to the cell receptor and therefore preventing the virus from entering the human cells and causing an infection. Now, monoclonal antibodies have been fairly recent technological advance, say over the last 10 to 15 years, but have been used for a number of diseases, both chronic diseases, immunological disorders, cancers, and more interest now in using these for infectious diseases. Now, as far as COVID-19 is concerned, there have been several efforts to develop monoclonal antibodies, and also there are different ways of doing it. You could do it in humanized mice, can also do it by extracting antibodies from people who have recovered and then purifying them, or it could also be done de novo in the laboratory. So different groups, different companies have taken different approaches. And at the moment, there are several antibodies, monoclonals, either used singly or in combination. So you can also use a combination of 2 or 3 antibodies. And this was something that was tested in Ebola and found to be very effective in a clinical trial done in the DRC. There were 2 different monoclonal antibodies that were actually found to be better than antiviral drugs in treating Ebola. So at the moment, we are aware of several clinical trials for the monoclonal antibodies. The NIH is sponsoring a number of these trials, looking at these both for prevention of infection. So you can give it to people who are been in contact, high-risk contacts, nursing home residents, etc., to see if you could actually prevent the infection. It's also being tested in early disease when people are not sick, outpatients, and also in those who've been hospitalized with more moderate to severe illness. So it's being tested across the spectrum, and it is one of the promising therapeutics on the horizon. I think the issue that you mentioned is very important, which is the possibility of scaling this and really making it accessible to people around the world, because it is a complex product to make. These facilities do not exist in all parts of the world. It could also be expensive, of course, depending on what technology is used. And therefore, we're looking very closely and also talking to many partners to see how technology transfer could be accomplished, and how, if once these are found to be safe and effective, just like vaccines, you also want them to have broader access. But it is a challenge, because technologically it is going to be difficult, but it can be done. And it would be useful in the long run to build this capacity in countries around the world, because monoclonal antibodies are likely to be therapy for other infectious diseases as well. And so this is a sort of platform technology that we really urgently need technology transfer to happen. Mariangela might want to add something to this.
Thank you, Soumya. Just to highlight, affordability is likely to be an issue because of capacity of production. Nowadays, the monoclonal antibodies that are already on the market for other diseases are extremely expensive. So they are among the high-cost drugs. So this is a concern. First, we have to have 1 or 2 monoclonal antibodies that prove to be safe and efficacious, and we need to have scale-up capacity. To that, let me say that through the Act A, the therapeutics pillar, we have been working already on the landscaping and the possibility to scale up capacity for some of these candidates who are proving to be more potentially effective and safe. The other issue that will come to light if we do have one of these drugs available for COVID will be also the flexibilities around the use of biosimilars that will potentially scale up the availability of the product in the world. So there are a lot of issues. It's very good, welcome news that potentially we have a good or more than one monoclonal antibody. The other side is we need to make sure they would be affordable and we would be able to scale them up. Thank you.
Thank you very much. No more. Oh, there is one more intervention.